Hi. In this video, we will study DNA replication, which is the process of DNA making a copy of itself to pass on to a new cell. The specific topics we'll talk about is the 4 required elements for DNA replication. We'll stress that DNA is always synthesized in the 5' to 3' direction, a fact which will cause some problems for DNA synthesis. And we'll discuss in detail the leading and lagging strands of DNA replication, which follows from the requirement that DNA is always synthesized 5' to 3'. So in today's lecture, replication, we'll talk about DNA producing DNA. In the next set of lectures, we'll talk about transcription, DNA producing RNA, and translation, RNA producing protein. These are words you have to memorize: replication, transcription, translation. On this slide, we see a double-stranded piece of DNA that's going to get replicated 3 times into 8 copies of itself. A key point here, which you've already seen, is that DNA replication is semi-conservative, meaning each of the daughter strands gets one of the original gold color strands, and it synthesizes a new, approximately dark red strand. And as you can see, each of the daughter cells from any parent has one of the original strands and also one of the new ones. On this slide, we see 4 requirements for DNA replication. Replication requires an origin of replication, the site for it to begin. It will not begin randomly at any place. DNA polymerase always requires a primer to get the strand started. That's a requirement of DNA polymerase. This is in contrast with another enzyme we'll meet today, RNA polymerase, which does not need a primer to start its synthesis. All polymerases need a template from which to copy, and all nucleic acid synthesis goes in the 5' to 3' direction. And we'll again see some of the challenges that causes for the replication machinery. On this slide, we see a growing strand of DNA in red here. Here is the end of the existing piece of DNA. This is the 3' hydroxyl group. Again, 1', 2', 3', 4', and 5'. The primes are important because those are positions of the sugar. The numbers without primes are for the bases. So you have the 3' hydroxyl group, a new nucleotide comes in, and it will allow the hydroxyl group to attack this first phosphate group. You're going to cleave pyrophosphate out, and you're going to have a connection between the 3' carbon and the new 5' carbon. This connection will only occur if you are bringing in the proper base to base pair with the existing base on the template strand. In this case, G is on the template strand, and C is the nucleotide that you're bringing in. Notice also that the template strand is anti-parallel to the growing strand, and that's depicted both explicitly with the labeling here, and also in this particular instance with the sugars being drawn upside down. If you were to bring in a base that didn't base pair with G, say an A, 999 times out of 1,000, the polymerase would not incorporate it here at the growing 3' hydroxyl group. Mistakes do happen very rarely, but most of the time they would get rejected. On this slide, you see the second of the 4 requirements, and that is that DNA synthesis begins at origins of replication. This is a piece of DNA that is defined by a specific sequence. The sequence is recognized by certain proteins that bind to replications of origin, and these proteins help unwind the DNA strands at the proper time, creating a bubble. By creating a bubble, you're going to expose bases and allow them to be replicated. One important thing to remember here is that there are going to be 4 replication processes. There's going to be a replication process going in this direction on this strand. There's going to be a second one at that position going in that direction. There's going to be a 3rd replication event going that way, and a 4th replication process going on at that end. So this is one replication bubble. There are 2 replication forks. Here's one replication fork, here's a second replication fork. And at each replication fork, both strands are being copied. So 4 simultaneous processes. Let's examine the growing replication bubble and start with just the leading strand. We'll talk about the lagging strand in a second. The first thing I want to point out is that we can deduce the directionality of the DNA because of the fact that the leading strand is growing in this direction. DNA grows 5' to 3', and DNA is anti-parallel. So if this is going 5' to 3' in this direction, this top strand is 5' over here and 3' over here. Since the DNA is anti-parallel, you know that this is the 5' end of this strand and this is the 3' end of this strand. And now let's imagine what's going to happen as this strand keeps growing. This strand will have to burrow into the center here to keep this unwinding, to keep copying the strand. And it will just keep going on for as long as there is a template to be copied. This motion is depicted in these 3 snapshots. The smallest bubble, a little larger bubble, and a larger bubble yet, just because as you keep unwinding at this location to keep copying the DNA, the bubble will get larger and larger as you're copying it. And as you can see for the leading strand, as the bubble gets larger, it just keeps extending and getting larger and larger. The same thing is true on this end. The leading strand on the bottom is just going, and it will just continue to make a very long strand as long as it is needed. The problem for the lagging strand is that it needs to be synthesized in the opposite direction that the replication fork moves. This is an unavoidable consequence of geometry. As this bubble grows larger, growing in this direction, on the bottom in the lagging strand, you are still required to synthesize in the 5' to 3' direction. So what happens is the polymerase jumps ahead and sequences back, jumps ahead and sequences back. We'll see that better on another slide. But the point here is just that a lagging strand has to put down many primers, and it can synthesize many small little pieces of DNA, which then later on will have to get stitched up. In this picture, we'll see half of a replication bubble, or one replication fork. The rest of the replication bubble would be here on this side. And as the replication fork moves out wider and wider, farther from the center of origin of replication, we're going to look at the consequences. Now, the book has drawn 2 figures that are not very related. Notice that down here we have 5' at the top, and here it is a 3'. So you'd have to flip this over for these 2 to relate. That's not super important. But here we have a leading strand synthesizing 5' to 3', and it's going to just continue to synthesize as long as you can go. The first amount of material that's synthesized is in dark red. Then we look at it a little later, and you can see that in lighter red or pink, there is additional synthesis, so that the most recently synthesized base of the leading strand is the sequence nearest the replication fork. For the lagging strand, again, it's a little more complicated. Remember, the center of the replication of origin is over here. The first synthesis is always closest to the origin. So this is the piece that was synthesized in the 5' to 3' direction. And then the polymerase jumps ahead to this one, and it's going to synthesize that one in 5' to 3' direction until it bumps up against this fragment. And then, as you see, these pieces are in dark red. There was a little more synthesis in pink right here until it stops. And then it jumps ahead again and is synthesized one more time in the 5' to 3' direction. This is one more slide of the growing replication bubble to emphasize that what gets synthesized most recently. You can always think of the oldest pieces of DNA in general as those that are at the center of the origin of replication. This is a symmetrical structure here, so these are the oldest pieces that are here. This one is the first part of the leading strand. Once again, the book has flipped the orientation, but that shouldn't bother you because you're getting good at looking at the 5' to 3' directions of things. And this is the first lagging strand, and this is an old piece of DNA. This is the first lagging strand on the other side. At some time later, the forks have moved out, and you can see in red what's been synthesized. So if I were to ask you, what is the last piece of DNA that has been synthesized in the lagging strand, you would go to the replication fork, find the first piece that you come to, and then you would go to the end. Here's the end. So here is also, by symmetry over here. Those are the last pieces of DNA that have been added to the lagging strand. So in this slide, we're going to look at some of the details of the lagging strand synthesis. We're not looking at the leading strand here, just the lagging strand. So this is just half of one replication fork. The replication fork would be moving in this direction, and the origin of replication would be over here. So as we've seen before in the lagging strand, you jump ahead and you synthesize back. Here we're synthesizing back, and then there'd be another step. We would have a primer here, and we would synthesize back because you always have to synthesize 5' to 3'. And now we can see some of the molecular details and some of the problems. First of all, why is some of this drawn in blue and some of this drawn in red? On this slide, DNA is in red and RNA is in blue. Now we're just replicating DNA, so we don't need any RNA to be there. And we don't, in fact, want any RNA in the final product. So what is it doing there? It turns out that RNA can be synthesized by an RNA polymerase enzyme that is called DNA primase, which is an enzyme that adds RNA to a DNA template. And this can be done without a primer, meaning that the first base that gets put down here does not need to have a 3' hydroxyl on which to attach to. It starts de novo. After a few bases of RNA, somewhere between 5 and 10, not a great deal, but now you have an established primer. And now you have a 3' hydroxyl group ready to be added to. And so DNA polymerase comes in, and it's going to add a DNA nucleotide to the existing 3' RNA hydroxyl group. And then it's going to continue to synthesize DNA. And it's going to synthesize it until it hits the previous RNA primer. And then it's going to chew away the fragments of RNA and lay down DNA. It's only going to stop when it hits the residual DNA, and until all that's missing is a phosphate group between the two sugars, or a bond between the phosphate group and the sugar. Then, an enzyme called DNA ligase will add that phosphate and make the connection between the phosphate group and the sugar, making it a complete strand of DNA. So for the lagging strand, we're going to use lots of RNA primers. We're going to synthesize a lot of short DNA fragments in reverse, and we're going to go until we hit RNA, chew it up, and keep synthesizing and stop again when we hit DNA. And then we're going to seal the nicks. And then the last thing that I point out, this is a term you have to remember. These short fragments of DNA on the lagging strand are called Okazaki fragments, after the scientist who first discovered them. This slide provides a little detail about the enzyme DNA ligase. It's a two-step process, and it takes a molecule of ATP, which is used for RNA synthesis. But it is also, as we mentioned, a high-energy intermediate molecule, which can be used to power different reactions. And the goal of the ligase enzyme is to seal a nick in the backbone. The first step is adding adenosine triphosphate. It's added to the existing phosphate group to activate the 5' end. And then, in a second step, that it's going to be released as you get bond formation. And now you have an intact backbone of this DNA molecule. So by hydrolyzing this high-energy intermediate ATP, we've paid the energetic price to do this otherwise challenging reaction of sealing the DNA backbone. So here's a more detailed picture of the replication fork. And here is that same picture, but an annotated version. So in this slide, we see a number of the proteins and the processes needed for DNA replication. Let's first talk about the unwinding step done by an enzyme called DNA helicase. This is going to unwind the DNA, which is needed for replication, since the two strands are twisted around each other. You might remember the old telephone landline cords, when they would get all twirled up, and that it would take some energy to pull the cord apart. That's required here at this replication fork. If you would just pull the strands apart without unwinding the DNA, then the part of the DNA ahead of you would get wound up even tighter, supercoiled, and eventually you would have a situation in which you could not continue. We'll see on the next slide how DNA helicase goes about unwinding the strand. If we look at the leading strand, DNA polymerase is just chugging along, adding new nucleotides in the 5' to 3' direction. DNA polymerase will bind to the DNA by itself when it's getting started replicating bases. And then a second protein called a sliding clamp, or pCNA, will attach to the polymerase. This protein helps lock the polymerase on the DNA so that it won't come off the strand prematurely, and more bases will be added. On the lagging strand, the action trails a little bit. Here's the polymerase with a sliding clamp, and it's synthesizing this way. Closer to the fork, we see the enzyme DNA primase, which is adding ribonucleotides to form an RNA primer, because it doesn't need the prior bases of a primer to do that. Eventually, this DNA polymerase will hit the DNA section, it will jump off, it will come back here, and it will start synthesizing down until it hits the RNA primer. It chews up the RNA primer and stops when it hits the already synthesized DNA. Now, single-stranded DNA is subject to various chemical and enzymatic processes. So single-stranded DNA is protected during synthesis by a protein called single-stranded binding protein. And it's going to cover the DNA until the polymerase gets there and starts doing its synthesis. Now, there's one last complication with this picture, which is too complicated to describe very well. So I recommend you look at the optional YouTube video on the edX site to see an animation. But briefly, in reality, this polymerase is physically located pretty close to the fort. The DNA is looped around so it will seem it's going in the same direction. This diagram that you see here is the unfolded geometry, which makes things easier to see. This is the link to the optional animation of DNA replication on YouTube, but it's also on the edX page. It's only 2 minutes, so I suggest you check it out. So for the last slide of this video, we're going to go back to the DNA helicase. Here's the polymerase in the leading strand, which is adding nucleotides to the growing strand. This is the newly synthesized strand here, and this is the template strand that is being read. And we're going to have to unwind the double strands before the replication fork moves ahead. In order to unwind this, the helicase relieves the torsional strain, working with an enzyme called DNA gyrase, by cleaving one of the DNA strands and then passing the cleaved strand around the intact one and resealing it. Gyrases and helicases are both part of a larger class of enzymes called DNA topoisomerases, which can alter the molecular makeup of the DNA, but they don't alter the molecular makeup, but they do change its topology, so topoisomerase, which is, in this case, the number of the times DNA is wound around itself. We'll take a break now, and then we'll go on to discuss how DNA is replicated at the ends of chromosomes. Thanks for listening.